Today we will discuss one important topic of plant physiology, that is photosynthesis. Our today's learning objectives are: we will discuss about historical background of photosynthesis, various discoveries and various statements given by various scientists, and then we will discuss about the pigments which are involved in photosynthesis. So we'll discuss photosynthesis. Photo means light, synthesis means formation. Plants can synthesize the chemical form of energy from light energy by using plant pigment chlorophyll, and to synthesize their food. That process is known as photosynthesis. That we all know that the source of energy in this earth's surface is sun. So plant use that solar energy to synthesize their food. They have a capability or capacity to harvest the sunlight and convert that into chemical form of energy. The term photosynthesis means that I've already discussed synthesis by using light. So what plants do, they convert sunlight into chemical form of energy, and that form of energy is converted into the organic material, and that organic material is utilized or used by other living organisms to sustain life on this earth's surface. And byproduct, they also you produce oxygen that is useful for all the aerobic organisms. So this is a very complex phenomenon, and the general reaction of photosynthesis is it involves 6 carbon dioxide molecules and 12 water molecules, which will results in the formation of this organic material, which is carbohydrate glucose in the presence of sunlight, and the pigment chlorophyll. So these are the two requirements for this reaction, and the byproducts in addition to glucose is very important, that is oxygen, which is evolved during this process, and water molecule. So these are the products of photosynthesis, and this form is utilized by all the rest of organisms directly or indirectly for all the activities. There are some bacterias which are chemoautotrophic, means they can synthesize their food by using certain chemicals like sulfur bacteria. So these bacteria use hydrogen sulfide in place of water, and their product is again the glucose, but they are not able to produce oxygen, they produce sulfur. So they show chemosynthesis, chemoautotrophic. They are chemoautotrophs, means they synthesize their own food by using certain chemicals. So there are some sulfur bacteria which show this phenomenon. In this picture we can see here that in plants their leaves contain one special cell organelle, that is chloroplast, which is also known as kitchen of cell. That chloroplast have photosynthetic pigments, especially the chlorophyll, which has capability to trap this solar energy and convert it into the chemical form of energy and synthesize their food, that we will discuss later on in detail. And the byproducts are oxygen and water droplets or water vapors by using this solar energy. And almost all the plants show this autotrophic phenomenon, means they can synthesize their own food. But there are some exceptions, like in case of angiosperms, there is one plant, Monotropa, that is commonly known as Indian pipe plant, that lacks chlorophyll, and that is saprophoric in nature, that use dead decaying organic matter for its growth. And there are some certain bacteria like sulfur bacteria and some protists like euglena that have the pigment chlorophyll, and they can synthesize their own food. And this photosynthesis results in formation of this organic material, which is used for the various life processes. Photosynthesis is the process which makes organic substance by using inorganic substance like carbon dioxide and water. And the byproduct is a carbohydrate, which is a glucose or blood sugar. So this is the process which begins all the food chains and food webs. Thus, we can say directly or indirectly, life is supported by this process, which is known as photosynthesis, and its byproduct is an oxygen gas that is important for all the aerobic organisms. This is the reaction by using carbon dioxide and water in the presence of light by using its pigment, that is chlorophyll. It can synthesize this organic materials. These are the inorganic materials. Carbon dioxide and water, it will synthesize this glucose molecule and also evolve oxygen that is utilized by the living organism for respiration. Next, we will discuss about the historical background of photosynthesis. The first viewpoint was put forward by Aristotle and Theophrastus. They believed that the plant absorbed all the nourishment from the soil. And our next scientist was Van Helmont. He concluded by doing some experiments that the dry weight of plant is because of water absorbed by plants and not due to the soil. Next was Stephen Hales, who is also regarded as the father of plant physiology. He discovered that the green plants require sunlight for the nutrition. Next was Joseph Priestley, who demonstrated that the green plants have capability to purify the foul air, that is phlogiston, produced by burning of candle. He has done one famous experiment which is known as bell jar experiment. In this experiment, he used bell jar, a mouse, a burning candle, and mint plant. He conducted a series of experiments. In first experiment, he took a mouse and a burning candle inside this bell jar so that no air can enter or leave this jar. And due to burning of candle and respiration of this mouse, they produces the foul air. And after some time, due to burning of this candle, the foul air was produced and the mouse died because of suffocation. So this was the conclusion, that this burning candle produces a foul air which is known as phlogiston. He didn't use other term, he used the term phlogiston for foul air. In next set of experiment, he placed a burning candle and a mint plant and mouse inside this jar, and he observed that the candle was burning and producing this foul air, which was purified by this green mint plant, and the mouse did not died because of suffocation. So he concluded that the burning candle produces the foul air, and that is termed as phlogiston, and this mint plant and the green plants have capability to purify that foul air, which and converted back into the fresh air, which is known as dephlogiston. So the green plant has capability to convert this foul air into the fresh air, which is known as dephlogiston. Later on, the term oxygen was used by Lavoisier. So this was all about this famous experiment of Joseph Priestley, that is known as bell jar experiment. Next scientist was Jan Eganhaus. He concluded that the green plants purify this noxious air during the daytime in the presence of light, and he proposed this equation, that is carbon dioxide and water combine to form organic matter, and they also produces oxygen gas. Next was Lavoisier, who identified that the pure air, that is dephlogiston, and we today know this by the name of oxygen, and the noxious air, that is phlogiston, carbon dioxide we can say, and other gases, other harmful gases which are produced during respiration. So these are produced by burning of candle as carbon dioxide. The next was Pelletier and Kevinton. They discovered the green pigment which is known as chlorophyll. So the discovery of chlorophyll was by these scientists. Next was von Mohl. He was the first to describe the presence of chloroplast in the plant tissue. Next was Deuterochet. He showed the importance of green pigment chlorophyll for the process of photosynthesis. Next scientist was Julius von Sachs. He demonstrated that the visible product of photosynthesis is starch. So starch is the reserve food material in case of plants, and showed that the chlorophyll is confined to the chloroplasts. So chlorophyll is confined or contained only inside the chloroplast. Next was Engelmann. He has done some famous experiments that we'll discuss in upcoming slide. Showed that the chloroplast was the seat of photosynthesis. That's why we call the chloroplast as kitchen of cell, plant cell, and discovered the role of different wavelengths of light on photosynthesis and plotted the action spectrum that we'll discuss in detail. Then there was scientist Blackman, who discovered two steps, that is light reaction and dark reaction. So photosynthesis occurs in these two steps, light dependent reactions or light independent reaction, that is also known by the name dark reactions in photosynthesis. He further proposed the law of limiting factor that we will again discuss in upcoming slide in next lecture. And there was a scientist named as Warburg. He performed the flashing light experiment by using a green single-celled non-motile algae, that is chlorella, as a suitable material for the study of photosynthesis, and he found that excess of oxygen is inhibitory for the process of photosynthesis. It means excess oxygen stops or inhibit the process of photosynthesis. Then next was Van Neal. He demonstrated that the bacteria uses hydrogen sulfide in place of water for the process of photosynthesis that we have discussed in first slide. Next scientist was Emerson and Arnold. They carried out flashing light experiments at different temperature and showed the existence of light and dark reactions that we will discuss in detail. Emerson's red drop effect in upcoming slides. Then next was Robert Hill. He performed the experiment that demonstrates that the main function of chloroplast is to photo-oxidize water, that is also known as Hill. So he was the first person to say that the water undergo oxidation and releases oxygen, and the source of oxygen is then water, not carbon dioxide in case of photosynthesis. Then there was Ruben Hassid and Kamen. They used this isotope of oxygen, that is oxygen 18, and confirmed that the oxygen involved in photosynthesis comes from water and not from carbon dioxide. So this credit goes to these scientists. The next was Melvin Kelvin. He gave the famous Kelvin cycle, that is also known as C3 cycle. They used the experimental material as chlorella and discovered that the first stable product of photosynthesis was a 3-carbon compound, which is known as 3-PGA or 3-phosphoglyceric acid. And they traced the path of carbon by using this carbon 14 isotope of carbon dioxide in photosynthesis, and they gave the Kelvin cycle or C3 cycle. Why C3? Because the first product of carbon assimilation was the 3-carbon compound, which is known as 3-phosphoglyceric acid, and was awarded Nobel Prize in 1961 for this discovery. And there was other scientists like Hill and Bandon. They proposed the Z scheme, which is also known as non-cyclic photophosphorylation, that we'll discuss under light reaction, and suggested that the two photosystem operates in series. There are two photosystems, that is photosystem 1 and photosystem 2. The next was Arnaud. He discovered the process of photophosphorylation. Photophosphorylation is the process of formation of ATP from ADP by using inorganic phosphate and light. Next was Peter Michel. He gave the famous chemiosmotic theory or hypothesis, which gave the idea of formation of ATP by using protons or proton motive force. Next was Park-Biggins. They discovered the quantosome. Quantosomes are the packets of energy, packets of lights, we can say. that helps in photosynthesis. And the size of this quantosome was 100 Armstrong thick and stated that it contains about 230 chlorophyll molecules. And there was coat shack. They discovered the formation of C4 dicarboxylic acid in sugarcane leaves. He found that the first product of photosynthesis in case of some plants like sugarcane was a 4 carbon compound. The next was Hatch and Slack. They gave the C4 cycle, that is also known as the Hatch and Slack pathway for carbon fixation, and they found that the first stable product of this photosynthesis was a 4 carbon compound which is known as oxaloacetic acid in some tropical grasses. So that process is also known as Hatch and Slack cycle. So this is all about the historical background and all the scientists which are related to this process of photosynthesis. Next we will discuss about the plant pigments. So plant pigments are divided into two parts. One are photosynthetic pigments and other is non-photosynthetic pigments. So photosynthetic pigments are those pigments which are directly or indirectly involved in photosynthesis. So they are again categorized into two parts. One is fat soluble pigments, means they are soluble in fat, and other are water soluble pigments. So firstly, fat soluble pigments are of two types. One is chlorophyll, that is the main pigment of photosynthesis. Then next is carotenoids. Chlorophylls are of various types, that is chlorophyll A, chlorophyll B, chlorophyll C, D, E, and bacteriochlorophyll that we'll discuss in detail in the next slide. And the other pigments are carotenoids, which are known as accessory pigments or helping pigments. Why helping pigments? Because those wavelengths of light which are not absorbed by chlorophyll, they are absorbed by carotenoids and they will give it to the chlorophyll. So carotenoids are again of two types. One is keratin, other is xanthophylls. They are differentiated on the basis of presence and absence of oxygen. So xanthophylls are the oxygenic carotenoids and these are without oxygen. So carotins are of different types like alpha carotene, beta cryptoxanthin, lutein, zeatin, and lycopene. So lycopene is the keratin which is found in tomato, and the red color of tomato is due to this carotene, that is lycopene, and beta carotene is the precursor for the synthesis of vitamin A. So जो vitamin A है वो बनता है beta carotene से, and this was first discovered in carrots, so that's why they are named as carotenoids or carotene. Next is xanthophylls. Xantho means yellow. So it is of again different types like zeaxanthin, neoxanthin, viloxanthin, and flavoxanthin. In addition to this, we have fucoxanthin also, that is also xanthophil. और जो fucoxanthin है वो present होता है brown algae में, that imparts brown color to the brown algae. The next category of pigment is phycobilins. ये class of pigment है plants में जो कि mostly lower plants में होता है, like in case of algae में भी red algae और blue green algae में. So this is the only class of pigment which is soluble in water. It is of two or three types. One is phycocyanin, which is present in BGA. We'll discuss it in detail. And next is phycoerythrin, that is erythrin means red color, so it is found in the red algae. Then we have a third category which is known as allophycocyanin or allophycoerythrin, that is also found in combination with these two pigments. Then we have some non-photosynthetic pigments that imparts color to the leaves, flowers, petals, and other plant parts, like anthocyanin, which are produced during autumn or winter season in the leaves. Then anthoxanthin, phytochrome, which is very important pigment related to flowering in plant that we'll discuss later on in our upcoming chapters. Then we have hematochromes that imparts a red color to the plant parts. So this is all about the pigment classification which are found in plants, and they are related with photosynthetic and some other activities. Next, photosynthetic pigments. Photosynthetic pigments mainly involves chlorophyll, carotenoids, and phycobilins that we have already discussed. It is chlorophyll's. So chlorophylls are of different types like chlorophyll A, chlorophyll B, chlorophyll C, E, and bacterial chlorophyll. So chlorophyll A, firstly we'll discuss about chlorophyll A. So this is the universal pigment which is found in all the autotrophs, and it is the chelate salt of magnesium. It is the chelate salt of magnesium, and the chlorophyll has a molecular formula as C55H72O5 and N4, and it contains central magnesium ion. So these four nitrogen molecules gets attached with this magnesium by forming the coordination bond that we will see in the picture, and its molecular weight is 893 Dalton. The structure has been established through the synthesis in the laboratory by Woodward in 1960. So the structure was given by Woodward, and its shape is of tadpole-like, and the molecule consists of a head and a tail. Head is of size 15 into 15 Armstrong and a tail of 20 Armstrong in length. The head is made up of porphyrin ring. Porphyrin ring is found by a pyrrole ring. Pyrrole is a heterocyclic compound that we'll see in this picture. So there are four pyrrole rings, so that's why this is known as tetrapyrrole. A closed ring derivative and the tail of phytol. And so head is made up of porphyrin ring, which is found by tetrapyrrole, and a tail which consists of this phytol, and the formula of this phytol tail is C20H39OH. So this is the picture of this pyrrole ring. So there are four pyrrole rings in case of chlorophyll head of this chlorophyll. So in this picture we can see here this one. So this is known as the head, and it contains four nitrogen, 1, 2, 3, 4, and these are four pyrrole rings, 1, 2, 3, and 4. So head consists of tetrapyrrole ring which are closed, and this tail consists of the phytol chain. This is the phytol tail which is formed by this long chain hydrocarbon. The chlorophyll A absorb blue, yellow, and red wavelengths of light and gives its peaks at 430, 578, and 662 nanometer respectively in the spectrophotometer. So this pigment is able to absorb these wavelengths of light. Next is chlorophyll B. The molecular formula of chlorophyll B is C55H70O6 and 4, and central magnesium ion is similar to that of chlorophyll A, and its molecular weight is 907 Dalton. It is similar to chlorophyll A except in having a formyl group, that is CHO group, instead of methyl group at carbon number 3. So this is the only difference in chlorophyll A and B. This is the structure of chlorophyll A. In this position, in chlorophyll A there is methyl group, while in chlorophyll B, in this position there is a formyl group, that is CHO group. So this is the only difference, otherwise they are similar. So it absorbs blue and orange wavelength and giving its peaks at 430, 595, and 644 nanometer on spectrophotometer. So it is found in all the green plants. So this is the pigment which is found in all the green plants along with chlorophyll A. So all green plants contain chlorophyll A and B, and except there are some blue-green algae and bacteria. Next is chlorophyll C. Its molecular formula is C35H32O5N4Mg, and its molecular weight is 712 Dalton. So this is the overall structure of chlorophyll C. It lacks phytol esterification, so phytol esterification is absent. It absorbs blue and orange wavelengths like that of chlorophyll B, and giving a spectrum peaks at 447 and 579 nanometer. It is found in brown algae, diatoms, that is Bacillariophyta, and in Pyrophyta and Cryptophyta. So these are some members of lower plants like algae which contain this chlorophyll C. Next is chlorophyll D. Its molecular formula is C54H70O6N4 and central magnesium ion, and the molecular weight is 895 Dalton. It has formyl group at carbon number 2, and it absorbs blue, yellow, red wavelengths of light and giving its peak at 447 nanometer, 548 and 688 nanometer respectively in the spectrophotometer. It is reported from the red algae. So this pigment is present in red algae by using some in vitro studies that we have found that chlorophyll D is present in case of red algae that we have already discussed. So this is the picture of chlorophyll D. Next we have chlorophyll E. So the molecular formula and structure of chlorophyll E is not clearly known. It has reported from some members of Xanthophyta, that is yellow-green algae like Vaucheria and Tribonema. So it absorbs blue and red wavelengths and giving peak at 415 and 654 nanometer. So it is found in Vaucheria. This is Vaucheria, that is yellow-green algae. It is a cenocytic. The next we have bacteriochlorophylls. So the molecular formula of bacteriochlorophyll A is C55H74O6N4Mg, having molecular weight of 911 delta. The structure is similar to chlorophyll A, and it absorbs ultraviolet radiations, yellow and red lights, giving peaks at 358, 391, 577 and 773 nanometer of wavelength. So this broad range of wavelengths can be absorbed by this chlorophyll, that is bacteriochlorophyll. And this is the porphyrin head of bacteriochlorophyll. It is found in all the photosynthetic bacteria. So photosynthetic bacteria have this chlorophyll that is known as bacteriochlorophyll, and the bacteriochlorophyll B is also found in some bacteria like Rhodopseudomonas, and the structure is not known. The next type of chlorophyll is chlorobium chlorophyll. It has hydroxyl methyl group and the carbon number 2 in the tetrapyrrole nucleus. So this is the group which is present in case of this chlorobium chlorophyll, and it is in heliophytes. Heliophytes are the plants which grow in sunlight, and the leaves possess chlorophyll A and B in the ratio of 5.5:1, whereas in sciophytes, that is shade-loving plants, the ratio is around 1.4:1 respectively. So that is the ratio of chlorophyll A and B in shade-loving plants and in sunlight-loving plants. Next, pigments are carotenoids. So these are the accessory pigments. The carotenoid are hydrocarbons with long aliphatic carbon chain which is joined together, and at the end they have two aromatic rings. So these are the carotenoid, that is the next class of pigment which are involved in photosynthesis, and they are sometimes called lipochromes. Why? Because they are soluble in fat and consist of 8 isoprene unit, and total number of carbons are 40. They are the main accessory pigments. There is helping pigment in photosynthesis and located within the chromoplast. So those wavelengths which are not absorbed by chlorophyll, they are absorbed by these accessory pigment, the carotenoids. They are found in non-green plant parts, and they are not necessary for their and the light is not necessary for their formation. So बिना light के भी ये जो pigments हैं, they can be synthesized in the plant, and the ratio of chlorophyll and carotenoid in shade leaves is 5:1. So 5:1, it means 5 chlorophyll molecule and 1 carotenoid molecule. The carotenoid pigments include two classes, that is namely carotenes, which are anoxygenic, means without oxygen, and other is xanthophylls that contains oxygen. So these are some essential carotenoids like lutein, alpha carotene, beta lycopene, that is found in tomato. The red color of tomato is due to this lycopene and zeaxanthin in the immature tomatoes. Next, carotenes. They were first isolated from the roots of carrots, so that's why they are known as carotenes, and they were first isolated by Wackenroder in 1891. Hence, they are named as carotenes. These are orange color pigments having empirical formula as C40H56 and molecular weight 536 Dalton. They are found in all groups of plant, that is from algae to angiosperms. Some of the common carotenes are alpha, beta, gamma, and delta carotenes, phytoten, lycopene, and neurosporin, etc. So this is the structure of carotene: alpha carotene, beta carotene, gamma carotene, delta carotene, eta carotene, and this carotene. So it is a long chain of hydrocarbons, that is, it consists of 40 carbons, and their ends contain two aromatic rings at each end, one aromatic ring at each end. So the lycopene is a red pigment found in the tomato, ripe tomato, and red pepper fruits. So pepper fruits also have lycopene. Then we have beta carotene, and this beta carotene on hydrolysis gives this vitamin A. Hence, the name carotene is also called as provitamin. Hence, this beta carotene is also known as provitamin A. Next, we have xanthophyll. These are the oxygenic carotenoids. They are yellow-colored carotenoids and are called xanthols or carotenols. So this is carotene, that is without oxygen, while xanthophylls, they contain oxygen, that is hydroxyl group. So the oxygen-containing carotenoids are more abundant in nature, and the ratio of xanthophyll to carotene is 2:1 in young leaves. So this is the ratio in case of young leaves. Xanthophyle formation takes place in dark, under aerobic conditions, so they don't require, so they require oxygen for the synthesis. The oxygen probably comes from the gaseous oxygen and not from water. So the gaseous oxygen helps in formation of xanthophylls. Next, the most common xanthophyll in green plants is lutein, and the brown algae, the brown pigment fucoxanthin that we have already discussed. The yellow autumn coloration of leaves that we see in winters is due to zeaxanthin, that is isomer of this lutein. Other common xanthophylls are cryptoxanthin, phylloxanthin, neoxanthin, etc. In bacteria, spiroxanthin is found, which has molecular formula similar to that of other xanthophylls. So in winters, we can see the yellowish color of this leaves are due to presence of this xanthophyll, which is known as zeaxanthin. So carotenoid mainly absorb violet, indigo, and blue wavelengths of spectrum, and to some extent the green wavelengths too, ranging between 400 to 505 nanometers. The maximum absorption occur from blue region. So these are some accessory pigments. They help in the process of photosynthesis by absorbing those wavelengths of light which are not absorbed by the chlorophyll. So that's why these are known as accessory pigments. The next function of carotenoids: first, they trap energy and transfer it to chlorophyll, because those wavelengths which are not absorbed by chlorophyll, they are absorbed by these carotenoids and they transfer it to chlorophyll A. Then they protect the photosynthetic apparatus from photooxidation. So excessive light intensity, the damage caused by excessive light intensity is also protected by these carotenoids, and they prevent the photooxidation of the chloroplast or chlorophyll. They prevent formation of reactive oxygen species. So these are some free radicals that cause oxidation in plant parts. So these reactive oxygen species are also prevented by, their formation is also prevented by these carotenoids. Next, we have third class of pigment, that is phycobilins. These are the only class of pigment which are water soluble. They consist of tetrapyrrole rings and supplementary accessory pigments. They are water soluble in nature. These are the structures of phycobilins. There are three classes of phycobilins, that is phycoerythrin, phycocyanin, and allophycocyanin. So the phycoerythrin is found in red algae, and this is found in BGA, that is blue-green algae. They are mainly found in red rhodophyceae and blue-green algae that we have discussed, that is cyanophylls. Then the prefixes R and C are added, which indicate their source, that is R phycoerythrin, C phycocyanin, like this. These are the prefixes which are used in case of biliproteins. Then the R phycoerythrin is the main pigment which is found in the red algae, the rhodophyceae, and C phycocyanin is found in the blue-green algae, and the allophycocyanin occurs in both the cases. So this is the red algae. So our phycoerythrin is found in red algae, and this phycocyanin is found in blue-green algae. The biliproteins also absorb different wavelengths of light. And phycocyanin absorb orange and green peaks at this wavelength, and phycoerythrin absorb yellow and green light. And allophycocyanin absorb orange and red lights wavelength of the spectrum. So this is all about this today's discussion. I hope you will understand some basic concepts of photosynthesis, and if you have any questions and queries, you can ask in the comment section, and you can give your valuable suggestion to improve so that I can improve myself. Thank you. Have a great day.